Hey guys, okay, in this lecture, we are going to now take a look at the second half of our chapter 4. We're taking a look now at bacteria. So let's take a look now. Okay, get in here. Here we go. So bacteria. So we're looking at, again, chapter 4. So chapter 4, but specifically bacteria. If you remember, I started last week's lecture by just showing you guys again why you guys are studying bacteria. Last week it was viruses. So I started off with an overview of some viral infections, and here's just an overview of some of your common bacterial infections. So for instance, sometimes Staphylococcus aureus can sometimes cause eye infections. Streptococcus pneumoniae is the specific bacteria that causes sinusitis. Streptococcus pneumoniae also can cause otitis media, which is an ear infection. Bacterial meningitis can be caused by Haemophilus influenzae, just all kinds of different types of bacteria that can lead to different things. If you look here, pneumonia can be caused by a variety of different types of bacteria when they get down into the lower lungs. Whereas then here are some specific sexually transmitted diseases. You guys probably have heard of chlamydia, gonorrhea. Those are some of your common sexually transmitted diseases. As I've mentioned, if you remember from chapter 1, I talked about one of our common indigenous microflora is E. coli, but it's also, as you can see right here at the top, our #1 cause of urinary tract infections. The E is Escherichia coli. There you go, cause of urinary tract infections. There are other types of bacteria that cause urinary tract infections, as your urethra is commonly, again, not just the urethra itself, but the upper part of the urethra as it gets closer to the bladder is typically pretty sterile because we see urine traveling through that. So anytime that bacteria gets up into the urethra, it can cause an infection. So there's just just an overview there. Again, I'm not going to quiz you guys on this particular picture. It's just something to kind of get you intrigued in looking at why we study bacteria and why we need to know it. So let's take a look. So bacteria can be divided into 3. When it says major phenotypic, it's what we see physically, what we're seeing physically in a bacterial cell. There's what's called gram-negative that have a cell wall, gram-positive that has a cell wall, and then those that lack a cell wall altogether. What does gram-negative and gram-positive mean? In short, if you remember, we talked about your pioneers of microbiology, and we talked about how Robert Koch helped to develop fixing and staining procedures for bacteria. And I briefly mentioned that a lot of bacterial cells are clear. So when we look at those through a microscope, remember light shines in through underneath, it's hard to see bacteria because it's just going to shine right through. So we have stains that are going to help to adhere to it's going to adhere to the bacterial cell itself to make it more visible. And then when I say fixing, there's a way that we can keep bacteria on a slide. A slide is a thin little piece of glass that's rectangular. And then we have slide covers that sit on top of that after we put stain on it, and that helps to keep it in place. So not that you have to know that, but when we talk about the staining, when bacteria are stained, they appear different colors. Sometimes bacteria show up, and you guys may have seen different photomicrographs in textbooks that you may have seen as an image of that obviously came from a microscope. And it may show up as either red, may show up as kind of a purplish color. And that is based on its characteristic. That particular bacteria will either develop more of a red stain, and that's your gram-negative. When you're seeing that reddish color, you're seeing gram-negative. Whereas if it's more of a purple color, you're seeing gram-positive. It's the way that the particular cell wall will take the stain, and that's what's happening there. There's different characteristics that make the gram-negative different than gram-positive, but that's all you guys need to know. Gram-negative typically shows up as red or a pinkish color, and gram-positive is a purple color. Your gram-negative cells are typically going to be the ones that are more resistant to antibodies, and that's going to make them a little bit more difficult to treat. Characteristics of bacteria, and we're going to see this as we go through our PowerPoint today, used in classifying your bacteria would include cell morphology. So we're going to see their shape, their staining reactions, which we just mentioned, the gram-negative versus gram-positive, motility, so meaning its ability to move, can be a very, wide variety of different ways that happens. Colony morphology, atmospheric requirements, so how it, either thrives in an oxygen environment versus a, non-oxygen environment, nutritional requirements, biochemical and metabolic activities, enzymes that the organism itself produces, its pathogenicity, and its genetic composition. So we're going to take a look at all of this today. So when I talk about morphology, I'm talking about its shape. So there are 3 categories of bacteria based on shape, and we're going to see a picture of these too. Your bacteria fall into, when it comes to what they look like, they're either going to be round shaped, and those are called cocci. If we're looking at just one of those singularly, it's called a coccus. So when we see multiple spheres or round shaped bacteria, those are called cocci. You're rod shaped, so those that are a little bit longer than they are wide, those are called bacilli. And then you have just either curved or spiral shaped, and that's the category itself. They don't have a specific name. Cocci sometimes will appear either by itself, or sometimes cocci sometimes get together, and we give them different names based on how they get together. So it's either seen by itself. If you see 2 cocci together, so just 2 round-shaped bacteria in a pair, that's going to be given the term diplococci. So the prefix diplo, so think di, meaning 2. If they appear in chains, that's where the term strep comes from. If you guys remember from that bacterial infections picture I was showing you guys, streptococcus is a prefix given to a certain name of bacteria, sometimes staphylococcus, so sometimes it appears in their name. But that's based on, again, how it appears on a microscope. We look on a microscope and we see these different round-shaped bacteria all in a line, in kind of a single file line. It looks like it's a chain. And so that's where we give it the prefix strep. So strep is the chain, so streptococci. If they appear in kind of a cluster or a group, so not in a single file line, but just all gathered together in a big cluster, that's where the prefix staph comes in. So staphylococci is a cluster. And then if they appear in packets of 4, we give it the term tetrad, or a packet of 8, we call it an octad. So remember, as we talked about when we were talking about microscopes, we said that the average coccus is about 1 micrometer in diameter. So that's how big that is, not very big at all. But again, still bigger than a virus. And again, as I mentioned, some cocci have the term coccus in their name. So when we see from that, again, from that bacterial infection slide I was showing you guys, remember that again, streptococcus or staphylococcus, when you see that in that name, that's where that comes from. It's how it appears on that microscope. So here again, just your general category of cell shapes. You have cocci, which are round, as you can see that there. Bacilli, here they're rod shaped. And then you have your curved and your spiral shaped, and they look just like that. In the next picture here, this is gonna further explain those cocci examples, what I mean, how if they appear by itself, it's just a cocci, just cocci if they appear just on their own. Whereas if they appear in pairs, you can see how they kind of, group together, just, they pair off, and like using the buddy system. Those are your diplococci. So diplococci, an example would be Neisseria gonorrhoeae, which is the type of bacteria that causes gonorrhea. So that specific type of bacteria tends to appear in pairs. So again, as scientists have just, over the years studied these different types of bacteria, here's what they're finding. Again, remember when we talked about Robert Koch, again, remember he was able to discover that bacteria and different things cause infections, and he developed what was called the germ theory of disease. So since then, years and years and years later, we have discovered, by studying, by looking at these different organisms under a microscope, we have eventually been able to identify them. So there's one example of how we can identify this one. Okay. So streptococci, remember, as you can see here, these are cocci that show up in chains. So see what I mean on how they're a single file line there? They all just show up as if they're, just getting in line. So streptococcus is in the name here. Streptococcus pyogenes is specifically the type of bacteria that causes strep throat. And then staph, again, cocci that appear in clusters. So see, they're not in a line here, they're just in a big group. So that's where staphylococci, that prefix staph comes in there. So you can see that in the name of this example. Staphylococcus aureus is the type of bacteria that causes boils on our skin. And then these two are a little bit less common as far as when it comes to infectious diseases. But tetrads just means that the cocci is showing up in four, in a pack of four. And then an octad is a pack of 8. So you can see there's 4 and then there's 4 right directly behind it. So in just a little group of 8 there. Okay, so here's some microscopic pictures that you guys can see here, and you can see gram-positive. You can see gram-positive Staphylococcus aureus. We just mentioned those are the ones that cause boils, but you can see they're gram-positive because they show up as kind of a purple color here, but see how they're showing up in those clusters. And then here you can see a scanning electron microscope image of Streptococcus mutans, as you can see that they're showing up in a chain there. Okay, and this is just a kind of more of an animated type example, but you can see the varied types. So here's some, actually we're going to see as we talk about bacilli in the next couple slides, they can show up in chains as well. Typically bacilli are not going to show up in clusters, so we're not going to see staph bacilli, but we are going to see streptobacilli. Okay, so let's take a look at that. So here are your couple spiral shaped, here are your rod shaped. This particular rod has a little tail coming out of it. We're going to talk a little bit about how that tail is going to give that specific bacteria its ability to move. So let's take a look. So bacilli is, again, we mentioned that's the rod-shaped bacteria. So oftentimes referred to as rods. They may be short or long. They may be thick or thin. They could have pointed or curved ends. But as long as they are longer than they are wide, they are referred to as bacilli. If they sometimes, just like our cocci, they can appear just by themselves. So they're just referred to as bacilli. But just like our cocci, if they appear in a pair, so two, so remember that two, we're going to see the same prefix, diplo. But these, again, are your rod-shaped bacteria, so we're seeing diplobacilli. If they show up in chains, same prefix, strepto, so streptobacilli. And again, like I said, there's no such thing as staphylobacilli. They don't show up in clusters. But that's what we're seeing there, just those two. Very similar to our previous slides there. So again, the average size bacillus is about 1 by 3 micrometers. Sometimes we have some extremely short bacilli that almost look even close to a cocci, but they're still a little bit longer than they are wide. But being so short, they almost are kind of hard to differentiate, so they're referred to as coccobacilli. So we're putting the names together there. These are just a couple different examples of your bacilli. The most common one that you'd probably be familiar with is Escherichia. As I just mentioned, E. coli is that #1 cause of urinary tract infections. That's going to be something we'll talk about a little bit later when we talk about hospital infections and how sometimes administering catheters, how sometimes that can happen. So we'll bring up E. coli again, but that's an example of a bacilli. Okay. And then when it comes to your curved and spiral shaped bacteria, you've got your examples of your curved shaped. So your Vibrio and your Campylobacter, typically these are food-based infections. Campylobacter is typically just food poisoning. But the most common one that you guys will probably come across is Helicobacter. In fact, on that bacterial infection slide I started off with at the beginning, if you looked under the area where it says gastritis, which is a stomach infection, the #1 cause for a lot of your stomach infections is caused by Helicobacter pylori, and Helicobacter oftentimes leads to ulcers. So anybody who's experiencing ulcers is probably has Helicobacter in their stomach. And some different spiral-shaped bacteria. Trey Treponema is an example of a type of spiral-shaped bacteria that causes syphilis. Borrelia causes Lyme disease, for instance. Not that you have to know specifics with these, but these are some good examples of just your different bacterial shapes here. All right, so here's one that shows the different spiral-shaped bacteria, specifically in a blood smear. You can see the different blood cells, and then your spiral-shaped bacteria. Okay, so as I mentioned before, when I showed you guys that picture that had that rod-shaped bacteria that had a little tail on it, and that's going to give your bacteria what's called its motility. Motility means its ability to swim, its ability to move. If something is motile, it's said to be able to swim. What is going to happen there? It's typically associated with flagella. That picture that I showed you guys, that little tail, that's what that's referred to as, is flagella. What that flagella does is it works very similar to how a sperm cell works. It works in kind of it kind of whips, and that's going to give it its ability to propel forward. So if it's, again, that term motile means its ability to swim, most often associated with flagella, which again is just a kind of a tail. Most spiral-shaped bacteria and about 50% of bacilli are motile. The spiral shape, typically the spiral itself can have the ability to move. And then those bacilli have the tail on it. So about 50% of your bacilli have that flagella on it. Most of the time, cocci, non-motile. Typically, they're going to stay pretty still. Bacteria can also be classified on their basis for atmospheric requirements, including their relationship to oxygen and carbon dioxide. With respect to that oxygen, bacterial isolates can be classified in the following categories here. So let me define these for you guys. What we're going to see, the difference between, and I'll talk first. I'll give you guys the term in front here in just a second, but the term behind you can see is either aerobe or anaerobe. In general, aerobe means oxygen requirement or oxygen requiring. If we think aerobe, think air, needs air. Think of like an aerobic activity. When a human does an aerobic activity like running or biking, we generally take in more air during that aerobic activity, and that's where that comes from. Whereas an, putting the prefix in front of it, this is without oxygen, is what that means. But let's take a look. There's a little bit more specifics when we add these terms in front. So the term obligate, if you think that, again, based on the term obligate, if we are obligated to do something, it means we have to do something, right? So an obligate aerobe is a bacteria that is only going to live in an oxygen-filled atmosphere. It is obligated to have oxygen, is a way that you can think about that. So in like our typical room air, like the air that we are breathing in as human beings, not that you have to know this number, is typically about 20 to 21% oxygen. You know, that's what we take in, that's what we need. That's what an obligate aerobe needs. Whereas a microaerophilic aerobe is still an aerobe, it still requires oxygen, but it is going to be okay with a little bit less oxygen. So that's where you can think of that term micro, meaning a small amount of, it can live in a smaller amount of oxygen. Okay. Facultative is kind of in between, is what this means. Anaerobes, meaning that it typically doesn't want to have oxygen, but it can tolerate oxygen, is a way to think about that. Or not, I shouldn't say tolerate, because that's going to be the next term there. Facultative, like I said, is kind of in between. They can live with or without oxygen, doesn't matter. Think about it that way. Okay. Whereas aerotolerant, again, that's where that term comes in, that this is an anaerobe, meaning it typically prefers a non-oxygen filled atmosphere. But again, it's going to be aerotolerant, meaning it'll tolerate it, but it doesn't prefer it. And then lastly, obligate anaerobe, again, opposite of obligate aerobe. These particular bacterial cells are going to require no oxygen. It's crazy to think about, because we think about human beings, and if we were in an atmosphere that doesn't have any oxygen in it, we aren't going to do very well, or we're going to die. But there are certain bacterial cells that can thrive in that atmosphere. And then the last term down there, capnophilic organisms grow best in the presence of an increased concentration of carbon dioxide, usually about 5 to 10% of carbon dioxide. Okay. And all bacteria need some form of element of, again, carbon, hydrogen, oxygen, sulfur, phosphorus, nitrogen. They all need a certain amount. So again, this is what we're talking about here, is that, again, we're talking about the classification of different bacteria, that some bacteria are obligate aerobes and they need a certain amount of phosphorus and nitrogen in order to thrive. That's one type of bacteria. So that's what I mean by this, that they all require certain elements. Some bacteria require, so elements themselves like calcium, iron, and zinc. Organisms with especially demanding nutritional requirements are said to be what's called fastidious. It's just a synonym here, as they give it the term. I always thought this was funny, fussy, because they are very specific with what they need in order to thrive. So again, as I just mentioned, the nutritional needs of particular organisms are usually the characteristics for that species and are sometimes important clues to its identity. So as scientists are constantly studying bacteria, again, like I said, based on, its need for these different elements and, these special minerals and things of that nature, that's going to give it a clue as to what bacteria we're looking at. Also, bacteria are classified according to its ability to produce its own, wastes and enzymes. So as bacteria grow, they produce many waste products, the same way that human beings produce waste products. If you know that as we go through digestion, we produce things like feces, we produce carbon dioxide when we breathe out, we produce sweat, we are excreting all kinds of different things. Bacteria work the same way. They go through, growth, and they produce waste products, some of which are enzymes. Pathogenic strains of bacteria like staphylococci and streptococci can be tentatively identified by the enzymes that they secrete, because typically it's these enzymes that may give it its ability to infect, to cause that type of infection. In particular environments, some bacteria are going to produce gases such as carbon dioxide and hydrogen sulfide. So again, we're identifying in a laboratory, we're identifying these different organisms based on what they're taking in, like carbon and nitrogen, calcium, and then based on what they're putting out, such as carbon dioxide. This is going to help us to identify these different bacterial organisms. To identify bacteria in the lab, they are inoculated into different substrates, meaning they're put into different, either different fluids or just different atmospheres. So such as with with carbohydrates and amino acids, and that's going to help to determine whether they possess the enzymes necessary to break down these substrates. Remember, enzymes we talked about in anatomy, enzymes that human beings possessed, we have lots of different kinds, but for example, like lipase in our saliva helps to break down carbohydrates in our mouth right away. So again, very similar. If a bacteria can break down carbohydrates, they probably possess that particular type of enzyme. So again, this is helping us to identify the different type of bacteria. Bacteria are also going to be identified based on its pathogenicity. So meaning its ability to cause infection. Remember the term pathogen, again, remember, is an infectious organism. So its ability to cause that infection. So many pathogens are able to cause disease because they possess a couple different things. One, some bacteria contain capsules. Think of a capsule as kind of like a shield. Like a capsule, when we think of like a capsule, like a medication, there's a capsule around that pill, right? And again, it helps, later when we digest it and it dissolves. But in this case, bacteria capsule is kind of like a shield that's going to protect it from being destroyed by the immune system. Pili. A way to think of pili, I always think of, I'm going to draw you another picture. I'm not an artist, I apologize. But with pili, we have a bacterial shape. This is a horrible rod-shaped bacteria. But pili are kind of like almost like little spikes. And that does a couple different things. One, protects it from the immune system. So it protects it from being destroyed. So it's going to be able to keep thriving and replicating and causing infection. But also those little pili help to adhere to the human cells that it's going to infect. It kind of works as kind of an attachment. So again, they're able to cause disease based on these numerous things. endotoxins and exotoxins. So we think of the word toxin, it's a bad chemical, a chemical that can destroy. And some bacteria have that ability to secrete a chemical that's going to be the exotoxin because it exits the bacteria. An exoenzyme, synonyms there, we're going to look at those that way. Just enzyme, meaning that it can break down something, and a toxin will destroy. But what these exotoxins and exoenzymes are going to do, they're going to damage cells. And that's what causes them to be destructive and cause disease in the human body. Frequently, pathogenicity is also tested in laboratory. What they do is they inject the organism into mice or sometimes cell cultures and see how that bacterial cell causes that infection and what it does. And these are just a couple different types of pathogenic bacteria. For example, Vibrio cholerae causes cholera. Not that you need to specifically know this, but for instance, this particular bacteria is pathogenic. It's the Yersinia pestis that causes the bubonic plague. So we're not seeing that obviously in our case. Hopefully you guys never come across the bubonic plague, but that is, again, a very pathogenic bacteria. Okay. All right, guys. So that, again, short and sweet with our bacteria lecture here. Just wanted to talk to you, just like I did with our virus lecture. I wanted you guys to just be familiar with, again, how we classify and how scientists are just studying these organisms in a laboratory and determining there's so many different viruses and so many different bacteria, how we determine that. And that's based on its shape, its need for oxygen, its need for different nutrients, its ability to cause infection, its ability to move if it has flagella. So all these different organisms can be classified, and that's going to help us to identify what bacteria that we're taking a look at. So that's what I wanted you guys to see here in bacteria. So very similar to what I did last week with our viruses, I'm going to post another video that may help us to give another visual that helps us to see bacteria and see what it's doing in the human body as it causes infection. And that helps us to understand bacteria a little bit further. But as usual, guys, if you all have any questions, just let me know.